Hi, Dr. Wang here. Today I'm going to do a training video for Plasmid Editor 4. We're going to use two sequence files. They're located on Moodle under this assignment. So we're going to actually use the same sequence files for assignment 4, 5, and 6. There are two sequence files in this folder. One is the PMSH2 sequence containing the wild-type version of MSH2, and another file is the PMSH2 plasmid that actually contain a mutated version of MSH2. So in this assignment, we are first going to use the ORF function to identify the open reading frame for MSH2, and then we are going to translate this DNA sequence to protein sequence for both the wild type MSH2 and the mutant MSH2 and compare the two protein sequences to identify the mutation. So that's question 2. So the first thing we're going to do is to download these two sequence files. And I have them already downloaded right here. So the next thing I'm going to do is to import these two sequences into the SAPIO platform. So over here, I'm going to create a new module and then drag the PMSH2 sequence onto it. All right, so here, first I'm going to rename this sequence. Click on this information icon to rename this as PMSH2. All right, and then I'm also going to open the second tab and then drag the mutant sequence to here. All right, so the second tab is right here. I'm going to rename this as PMSH2 mutant. All right, so, and I'm also going to rename this module as 4. Right, so in this activity, we are going to use the ORF function and the alignment function. All right, so now everything is ready. We are first going to identify the open reading frame. Actually, this is a circular DNA molecule, so I'm going to make sure that I'm looking at a circular molecule first. The ORF function, actually, we have done this right here. Right, so we know that PMSH2 plasmid contain the coding region for gene MSH2, which is a little more than 900 amino acid long. All right, so therefore, there is an easy way to identify this open reading frame. So I'm simply going to go to ORF detection, starting from position 0, right, the very beginning, and make sure that I'm searching for an ORF that's at least 900 amino acid long. This, of course, only applies to MSH2 because I already know that this protein is quite long. So after scanning, we can see that the software has identified this open reading frame, right? So I'm going to click on this arrow to select the sequences and then coming back to the translation function. So you can see that this is on the forward strand. Right, it's in a clockwise orientation, so it's on the top strand or the forward direction. So I'm going to use translate the forward direction. So here, this translation gives a protein sequence that's this many amino acid long. So this number is the answer for question number 1, right? So this protein contains this number of amino acid. So the next thing I'm going to do is to copy this protein sequence. So actually on Mac, Command C doesn't work. So you will have to go to the browser and then choose copy right here. All right, so I have copied this protein sequence. The next thing I'm going to do is to do an alignment. So the alignment function is right here, right? So if you click on this button under analysis, there is a function called sequence alignment. Clicking on that will open a new tab called sequence alignment. You can, of course, align DNA sequences. That's right here. But I want to align protein sequences, right? So here I'm going to use this window on the right, paste DNA or protein sequence. So I'm going to paste the protein sequence that I just copied. And then this is the MSH2 wild type protein. All right. So I'm now going to return to this tab for PMSH2 mutant sequence. So the first step is again an ORF detection, starting with the very first location, searching for something that's at least 900 amino acid long. So, and this has identified actually the same ORF. Okay, so I'm going to click to highlight the sequences and then use the translation function to translate in the forward direction. So over here, you can see the mutant protein actually is the same, has the same length. It's just the sequence itself varies slightly. I'm going to again copy the mutant protein sequence, copy that, and then come back to the alignment tab, right? So here, scroll down and paste it into the second window. And then I'm going to call this MSH2 mutant. Okay, so this is the sequence for the mutated MSH2. So down here, I'm going to tell the software that I want to align two protein sequences. And then that's it. I'm going to run analysis. So here, the wild type sequence and the mutant sequence are lined up. You can see that the two sequences are very similar to each other, except a difference that's right here. So in the wild type, right, so in the wild type at this position, we can see that the two sequences that are actually identical, except this single mutation. So we normally name mutation starting with the amino acid in the wild type. So this is an isoleucine, right? So, and then the middle number should be the number or the location of this amino acid. So this is the 141st amino acid, right? And then it's mutated to methionine. So, right, so isoleucine or I at position 141 mutated to methionine or M. So this is the answer for question number 2. So which mutation, which of the following, right? So we can see that the way we describe mutation is the first letter represent the amino acid in the wild type. The number in the middle represent the location of this amino acid, and the last letter represent the amino acid in the mutant sequence. All right, so this is how to do this assignment.